Welcome to Cancer Lesson 2.4, How are cell activities controlled in a normal cell and disrupted in cancer? The goals of this lesson are to introduce how cells that have the same DNA can perform different functions because they express different genes, and consider how changing gene expression can result in uncontrolled cell growth. At the end of the lesson, the students should be able to explain how different cells can perform different functions even though they carry the same DNA, explain the term differentiation, and be able to describe the roles of stem cells and terminally differentiated cells, explain how gene expression is controlled by transcription factors, and explain how gene expression is also controlled by the opening and closing of DNA. We will achieve these goals by engaging in a Socratic discussion and an interrupted case study on variations in gene expression in different cells. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. All cells in an organism have the same DNA, but can perform different functions because they transcribe different genes from it. Gene transcription is regulated by whether transcription factors can access the gene's DNA and changes as a cell acquires its mature function. The process of cell maturation or specialization is called differentiation, and functionally mature cells are said to be terminally differentiated. You can review the scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. The key points of the do now are that all cells in an organism have the same DNA, but because they express different genes, they express different proteins and therefore have different structure and function. Stem cells are embryonic cells that have not yet begun to specialize. When stem cells divide, they either replicate themselves or make cells that are beginning to specialize structurally and functionally. This specialization to acquire a mature function is called differentiation. We illustrate structural and functional specialization using three different types of cells: muscle cells, neurons, and lung epithelial cells. All of these cells contain the same DNA, yet they look so different. This is because function is related to structure, and structure is related to the proteins the cell makes. So the muscle cell is making muscle proteins, but not neuronal or epithelial proteins. But all the cells in an organism arise from a single zygote, so how does this structural and functional specialization occur? We use an analogy to describe how cells eventually gain different functions. Much like all babies in a community seem more or less identical when they are born, but they each eventually develop into adults with different jobs, the first stem cells that form from a zygote are identical, but eventually they specialize. They gain new structures and functions and end up as one of the many different cell types in the organism. This type of specialized replication is called differentiation. But how exactly do cells become specialized? The key points of the discussion are: cells differentiate by expressing the genes for proteins that permit specific structures and functions. To express these proteins, specific transcription factors need to attach to the genes and initiate gene transcription. Transcription factors can only access genes whose DNA is not tightly coiled. The status of a gene's DNA is called its epigenetic state. Differentiation occurs as cells acquire increasingly specialized characteristics. For example, a columnar epithelial cell in the lung is more specialized than a columnar epithelial stem cell, because the columnar epithelial stem cell could also become a columnar epithelial cell in the esophagus. The more specialized a cell is, the less likely it is to divide, because each time it divides, it has to break down and reform the structures that make it specialized. Hence, many terminally differentiated cells lose the ability to divide. In the same way that most very successful lawyers cannot just become very successful artists, terminally differentiated cells normally can't turn into functionally different cells. For example, a neuron won't just turn into a muscle cell. Of course, a lawyer could become an artist if she acquires art skills, but the more specialized a cell is, the less likely it is to be able to become a different type of cell. How does specialization occur if all the cells have the same genes? Cells are different because they express different genes. The bars in this diagram represent the genes in each cell, and the blue color represents whether the genes are expressed. For example, the red blood cell, muscle cell, and the pancreatic cell all have the gene that codes for hemoglobin, but only the red blood cell expresses it. And this is key. The reason why cells specialize is because different genes are expressed in different cells. So what controls which genes are expressed? Gene expression is controlled in two major ways. First, through proteins called transcription factors that attach to the gene and actually initiate expression of that gene. A gene won't be expressed unless the cell has made that transcription factor. Second, through the state of the gene's DNA itself. The transcription factor can only access the gene to attach to it when the DNA is uncoiled enough. The open or closed state of the DNA is called its epigenetic state. The next slide briefly reviews molecular dogma. Cell structure and function depends on proteins, but how are these proteins made? DNA, which is located in the cell's nucleus, contains genes, which are the instructions for every protein a cell could make. The DNA code is read, much like a book, and transcribed into a molecule called RNA. This RNA is then read by ribosomes, which translate, much like translating into another language, the information in the RNA to create a particular protein. Each cell in an organism has identical DNA. Different cells are specialized in different ways because they make different proteins, as illustrated here. This is called gene expression. But how do cells control which genes they express and when? Genes are only expressed when proteins called transcription factors attach to the start of the genes and initiate transcription of the gene. Different genes have different transcription factors. But how do these transcription factors get access to the genes? Students may recall that DNA is a double helix that is tightly wound up in the cell's nucleus. If you uncoiled it, it would stretch for nearly 2 meters. The DNA is coiled around proteins called histones, much like a string can be wound around a spool. Using this analogy, the string is the DNA and the spools are the histone proteins. A transcription factor can't access a gene if the DNA is tightly spooled up. The gene needs to be uncoiled from the histone protein so that the transcription factor can attach to it. The reason DNA coils up tightly on histones is because DNA is negatively charged and histones are positively charged, so they attract each other. But when areas of DNA are modified with small molecules called acetyl groups that are also negatively charged, they repel the DNA's own negative charge so it can detach from the histone and uncoil. The transcription factors can then worm their way in and attach to the genes. But what if we don't want to express certain genes? In this case, small molecules called methyl groups attach to the DNA, and because these are positively charged, they attract the DNA and make it coil up and attach to the histones more tightly. So now the transcription factors can't access these genes. Now we can see that a lawyer may have difficulty becoming a successful artist if the genes that confer artistic skills have been switched off. This can occur if the DNA in these genes has been modified with methyl groups that has caused the DNA to coil up tightly, making them inaccessible to transcription factors. In the activity, students will work in small groups to complete an interrupted case study on variations in gene expression in different cells. The key points of the lesson's activity are that acetyl groups uncoil DNA and methyl groups coil up a gene's DNA, regulating its epigenetic state. The epigenetic state of DNA in cancer cells is generally more uncoiled than in normal cells, so genes that are normally shut off can still be expressed. In the activity, students examine the epigenetic state of various genes in different cell types. They examine the relationship between epigenetic state, gene expression, and the function of the cells. Finally, they compare the epigenetic state of certain genes in cancer cells compared with normal cells. The key points of the lesson's wrap-up are that epigenetic regulation controls how open or closed DNA is. Acetylation involves opening up DNA. Methylation involves closing up DNA. In cancer cells, DNA tends to be more open. Genes may be transcribed even when they should not be. And terminally differentiated cells may gain the ability to perform other functions. Whether or not a gene can be transcribed depends on its epigenetic state. Whether it will be transcribed depends on the transcription factors present. Healthy cells switch gene expression off by attaching methyl groups to areas of DNA so that it is tightly coiled on the histones and inaccessible to transcription factors. But in cancer cells, the overall epigenetic state of DNA is more open, which makes genes more accessible to transcription factors. Students should think about what happens if the gene exposed is a proto-oncogene. Normally, differentiation goes in the direction of increased specialization, with terminally differentiated cells losing the ability to replicate. But in cancer cells, differentiation can sometimes go backwards, so terminally differentiated cells behave more like undifferentiated cells, regaining the ability to replicate because certain areas of the DNA become more open. For homework, have students read the 2.4 homework worksheet on the stem cell theory of cancer. This theory proposes that cancer stem cells arise out of our normal stem cells. They should also read the workbook chapter 2.4 and answer the worksheet and workbook questions. One of the most common questions students ask is what happens if a key gene is mutated in a cell that normally doesn't express that gene? There will be no effect on that cell. However, cells that do express that gene will be affected. This is why we have tissue-specific cancers. Another question students ask is, are there any mutations that can lead to cancers in many kinds of tissues? Yes. If genes that control the cell cycle are mutated, such as RB, which we discussed in Lesson 2.2, then many cell types will be affected because checkpoint proteins like RB are common in every cell. Therefore, RB acts as a hub. Recall that we talked about hubs in Lesson 2.3. At the end of the lesson, collect the lesson worksheets to assess students' explanations of epigenetic changes in cancer cells. This lesson introduced the concept of how gene expression is regulated in normal cells and cancer cells. We will build on this concept in future lessons to understand how normal cells become cancerous. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.